Hello everyone, welcome back. So the next topic we're going to be talking about is microbial metabolism and this very cyclic nature of microbial metabolism as far as putting energy in to build things and then breaking things apart to release energy. And really it all comes down to energy. Living things need energy in order to go through all of the processes and do all the necessary things to be a living organism. And so microbial metabolism is talking about getting energy from a source and then using that energy to do something. This diagram right here gives you an overview of all the biochemical pathways involved in microbial metabolism. And our metabolism, they are very similar. So by the end of this lecture, you should have a better understanding of what's kind of going on in this diagram, how you get from point A to point B, and so on. If this diagram looks very overwhelming now, don't worry about it. We're going to go through step by step and really understand the processes involved in this diagram. The big takeaway, though, and what to keep in mind as you're going through this lecture, is the big idea is that we're starting with a molecule of glucose that has a lot of energy built up in it, and we're going to break it down. We're going to keep breaking it down further, breaking it down, and getting energy out of that glucose in the form of ATP. So every time you see ATP, adenosine triphosphate, that is energy. So we're breaking down glucose to get energy. Before we dive into those specific processes involved, we're going to talk about kind of the overview of what's going on and these underlying mechanisms that are necessary in order to allow those different processes to happen. So the first one we'll talk about is catabolism and anabolism. Catabolism and anabolism is really referring to that cyclic nature I talked about in the beginning, the building and the breaking down. So anabolism is building larger molecules. So it's taking small molecules and using energy to build larger molecules. You have to put in energy to build. Catabolism is breaking down large molecules to release energy. So it's taking large molecules, breaking them down into smaller molecules, and in the process, releasing ATP or energy that can be used for other things that the cell can do. And so the process of breaking down glucose to release energy is a catabolic process. We're taking the large molecule of glucose and breaking it down into smaller molecules and releasing ATP from that glucose. But just keep in mind that metabolism is this collective of this building and breaking. So metabolism isn't just breaking down glucose. It's the whole collection of all those processes in the cell in the breaking and building of these molecules. Another slide kind of restating what I said on the previous one. But just remember that anabolic reactions require energy. And this is building a more complex molecule or more complex material from a simple substance. An example of this would be the formation of proteins. So you're building proteins from amino acids. Amino acids are the building blocks of proteins. Amino acids are being stuck together, built together in order to make a protein. And that requires the input of energy to stick those amino acids together. Catabolic reactions release energy, so some complex molecule is broken down into simpler ones, and this releases energy. That energy is stored between the bonds of that complex molecule, and as those bonds are broken, it releases energy that can then be harnessed to do something else in the cell. Of course, the example here would be breaking down glucose or sugars through those processes that ultimately ends up in the release of carbon dioxide and water as the kind of the final outcome of the breakdown, and also energy, of course, that the cell harnesses. A fun way to kind of remember this that I heard from one of my students a previous semester is that catabolic is break because cats like to break things and knock things off of shelves and countertops. So catabolic is break. Now on to the next topic. So we know that building and breaking is an important part of metabolism, and now we're going to talk about oxidation and reduction. And this is really important because this is how energy is kind of moving throughout the metabolic process. So there's a lot of energy stored in something like glucose. And so us and bacterial cells and microbes want to get energy out of those bonds because energy is stored within all of those bonds of glucose. And we ideally don't want to just break apart glucose and lose a bunch of the energy. So we want to break it down incrementally and store that energy as those bonds are broken, move it through the metabolic process, and ultimately get a lot more energy out of it at the end. How are we going to accomplish that is through oxidation reduction reactions or redox reactions. And redox reactions really complement each other because we're talking about the loss and gain of electrons. And so when one compound loses an electron, another one gains an electron. So oxidation is the loss of electrons. Reduction is the gain of electrons. And a helpful memory tool to remember this is oil rig. Oxidation is loss and reduction is gain. And you can see here these two images. You can think of this in terms of electrons and hydrogens. So over here in terms of electrons, compound A lost an electron, it was oxidized. Oxidation is loss, it lost an electron. Now it has a positive charge because electrons have a negative charge, so it lost that negative charge and became positive. Compound B gained an electron, it now has a negative charge, and it was reduced. Reduction is the gain of electrons. So you can see how this happens in tandem. One compound was oxidized, while the other was reduced, and A gave an electron to compound B. So as these bonds are broken, we're releasing energy and storing it in the form of electrons by moving these electrons and these bonds from one compound to another. You can also think of it in terms of hydrogens because hydrogen has one electron and one proton. And as we'll see on the next slide, the electron carriers that we're talking about are most often talking about removing a hydrogen or adding a hydrogen. But that's the same concept that's happening down in this image. So you have this oxidation reaction where you start out with a compound with these electrons. It gave electrons to this electron carrier, which we'll talk about on the next slide. But it gave these electrons away, and now it is oxidized. Oxidation is the loss. Our electron carrier took those electrons away, and so at the end it was reduced because it gained electrons. So now we know that energy can be shared and between different types of things in the form of electrons, but what is going to actually be doing that job in the metabolic process? And that is electron carriers. So electron carriers, as the name implies, carry electrons. And by carrying electrons and forming that bond to carry that electron, that is potential energy. So they're storing potential energy in that bond, and then they're taking it through the metabolic process to go use that energy somewhere else along the process, as we'll see later. So energy is stored in between that bond. Whenever an electron carrier makes a bond with a hydrogen, energy is stored in that bond, and that's what it's moving along through the metabolic process. Here are some important electron carriers that are in charge of moving energy throughout metabolic processes. So we have nicotinamide adenine dinucleotide, so that's NAD plus to NADH, nicotinamide adenine dinucleotide phosphate, or NADP to NADPH, and flavine adenine dinucleotide FAD to FADH2. And I will not be asking you for these names, you just have to know their acronyms over here. And really make sure that you understand their oxidized and reduced forms. So up here we have NAD plus and NADH. So as you can see, we're looking, it's kind of like one of those games where you try to figure out what's different on each side. But if you look right here, there's one hydrogen right here. There are two hydrogens right here. So NADH gained a hydrogen or gained an electron. That must mean that NAD plus is oxidized. Oxidation is the loss. It lost an electron in this form. Reduction is the gain, so that means NADH must be the reduced form. It gained an electron in the form of a hydrogen. So this is the potential energy form. This energy stored right here with this hydrogen bond. NADH has potential energy, and it's going to go use that somewhere else along the metabolic process. On to our next topic. So now we know building and breaking is important for metabolism. We can see how energy moves through the processes of metabolism by shifting electrons from one carrier to another. And now we're going to talk about enzymes. And enzymes are essential for metabolism because if we didn't have enzymes, metabolism wouldn't happen. It wouldn't happen for us, it wouldn't happen for bacterial cells. Enzymes are an essential part of making sure metabolic processes happen. So enzymes are made of proteins. And isn't this a beautiful image of a ball and stick model of the enzyme hexokinase? It's just gorgeous. They're very complex and they're very specific, as we'll see. Enzymes are also catalysts, so they increase the rate of a chemical reaction. So they make that reaction happen more readily and more quickly. And they do this by having very specific active sites that bind to specific substrates. So an active site on an enzyme is where a substrate binds, and they're very specific to a certain type of substrate. And this is what makes that reaction happen more quickly. So enzymes essentially help two substrates meet that would normally take forever to meet each other. But enzymes make those two molecules meet and then make a reaction happen way more quickly than it would if the enzyme weren't around helping being a matchmaker for these substrates. So like I said, they increase the rate of a chemical reaction by lowering the activation energy. And we'll see more about this on the next slide. So this graph is showing you the importance of having an enzyme based on the amount of energy required or the activation energy required to make a reaction happen. So we have two reactants over here, reactant A and reactant B. You can see reactant A requires way more energy, a much higher activation energy for this reaction to happen and for it to become the final product. Whereas reactant B, it requires half as much energy. The activation energy is half as much and in the presence of an enzyme in order to turn into that final product. And something that I do want to mention here is that enzymes are never used up. So once an enzyme helps two molecules meet, it'll go off and it'll help two more molecules meet. Very, very specifically. Enzymes have one job, they help one reaction take place, and that's it. They're not multitaskers, they have one job and they do it over and over and over and over and over again. So they don't get used up and they're actually not part of the reaction, they just catalyze the reaction and then leave and go help someone else. Now, something else, enzymes are really important in living things because you don't have to change anything about the environment to make this enzyme do its job. It's in this kind of optimal environment where it's going to help these molecules meet again and again and again. But if this wasn't happening in a living organism, maybe in a test tube, a way that you can lower the activation energy or increase the rate of reaction is by increasing the temperature. So this makes the molecules get all jittery, they move around more quickly, they're more likely to bump into each other. So this lowers the activation energy. And you can also increase the substrate level. So you can dump in a bunch more substrate so they're more likely to bump into each other. But those two options do not work in a living organism. We have a very specific homeostasis, and so do bacterial cells. So you can't throw off the temperature or the substrate concentration. That's why enzymes are essential for making sure metabolism happens in living organisms. So I like this image because it really shows you again how specific enzymes are. So in this example, we have the enzyme hexokinase that's catalyzing the reaction where it's using energy to turn glucose into glucose 6-phosphate. And just so you know, I will not be asking about any of these specific molecule names at all. We're thinking more big picture here, but this is just to show you as an example. So hexokinase has these very specifically shaped seats where it will allow one glucose molecule to sit down on the enzyme, and it will allow one ATP molecule to come sit down on the enzyme. And these seats are so perfectly and cozily arranged on the enzyme that it perfectly aligns this phosphate molecule right next to glucose. So glucose will just pop off that phosphate molecule, take it away, and become glucose 6-phosphate. So hexokinase, this enzyme, brings these two molecules together and allows this reaction to happen really efficiently with these perfect little seats. And as you can see, it's not part of the reaction up here. You don't see hexokinase in the reaction. It just helps catalyze the reaction. And you can see these very specific active sites are specific for glucose and ATP. So other molecules aren't going to come use this enzyme for anything. Finally, I want to mention something about this word down here, hexokinase. So in many cases, enzymes you'll see end in ase. Hexokinase, ace. So keep that in mind for the future. It's just a nice thing to look out for if you're trying to identify what is an enzyme. Most end in ase. Now we know what an enzyme does, but what is an enzyme made of? So an enzyme has a portion called the apoenzyme. And if you remember, enzymes are made of protein, and this is that protein portion that is folded into this specific 3D shape that allows it to be highly specific for what it's binding with. So the apoenzyme is usually not active or an inactive form until it's paired up with a buddy called a cofactor or a coenzyme. So an apoenzyme needs to be bound to a cofactor or a coenzyme to be active. And a cofactor is a non-protein component, the same as coenzyme, it's a non-protein component. But the cofactor is going to be an inorganic compound, maybe like iron or zinc. But then the coenzyme is an organic cofactor, and we're going to be talking about a few important coenzymes as we go through the metabolic process. We're going to talk about coenzyme A, and other examples are NADH and ATP can also act as coenzymes. So an apoenzyme binds with the cofactor or coenzyme and then becomes a complete and active enzyme called a holoenzyme, and that's the final functional enzyme. You can see in this graphic right here, the inactive enzyme or this apoenzyme portion has to be bound to its coenzyme in order to be active and actually bind the substrate. So I feel like this is a really clear image showing how important it is for the inactive portion of the enzyme to be bound to its cofactor or coenzyme. This is again showing the relationship between an inactive protein portion called an apoenzyme and its cofactor. So this would be considered a conjugated enzyme because two parts have to come together to make it active. There are also simple enzymes that don't need a coenzyme or cofactor. They're active by themselves, but holoenzymes require this cofactor or coenzyme. And you can see this protein portion has this very specific shape. And this is useful for just showing you that, remember, enzymes are made of proteins. Proteins are made of amino acids. And all those amino acids fold together to make a very, very, very specific shape. And in this case, this specific shape needs to be bound to a specific coenzyme. And then those two together create a holo or complete active enzyme. And it also creates this very specific shape that allows the binding of the substrate. So it's all of those levels of specificity in terms of the three-dimensional shape of the protein portion and binding to its coenzyme or cofactor that allow it to be an active enzyme and actually bind the substrate. Now we know enzymes are specific. They bind very specific things. So enzymes are pretty picky, right? Because they also require very specific optimal conditions in order to function, which is fine because those optimal conditions are the organism or the environment where that enzyme normally is. But if you alter the temperature, pH, and substrate concentration, then you can alter how that enzyme works and really render it completely ineffective. So enzymes are made of proteins, and proteins are made up of chains of amino acids. So think back to this ball and stick model of hexokinase. Look how complicated that is. So this is all held together by different types of bonds, like hydrogen bonds. When the temperature is too high, those hydrogen bonds that are keeping this 3D structure together just fall apart. So the protein that has this beautiful 3D structure, its active form, it denatures. Those hydrogen bonds break, and then it's this denatured protein that no longer has a function because it doesn't have that three-dimensional structure anymore. The same thing with pH. When pH is altered, then it messes with the hydrogen bonding in that 3D structure and renders it ineffective. For substrate concentration, you only have so many enzymes in a living organism, so you can add a bunch of substrate in, but you're going to oversaturate the enzymes. They can only do so much work at once. And then finally, what we're going to talk about over the next few slides is inhibitors. So enzymes can also be inhibited a few different ways, and we'll talk about these over the next few slides. So the first one we'll talk about is competitive inhibition. This is one way that we can inhibit an enzyme. And make sure you can identify these different structures. So we have the actual enzyme. So this is made of protein, remember. It might have a cofactor or coenzyme present. It doesn't show it on this slide. But this is made of protein. It has a specific 3D shape that provides this specific 3D shape for the active site where the substrate will bind. For competitive inhibition, we also have a competitive inhibitor that looks a lot like the substrate. It has a very similar shape. And this competitive inhibitor will compete with the substrate to bind the active site. So it's such a similar shape, it can actually bind this active site and block the substrate from binding, making this enzyme completely ineffective. An example of this is a drug called trimethoprim. So trimethoprim actually blocks the folic acid pathway for bacteria. It introduces a competitive inhibitor that looks a lot like folic acid, and so those analogs, those folic acid analogs, will block the active site of the bacteria's enzymes. The bacteria can no longer go through that metabolic pathway, and it dies. On to non-competitive inhibition, another way to inhibit an enzyme. So for non-competitive inhibition, the inhibitor is no longer competing for the active site. So it's not competing for the same site. For non-competitive inhibition, the inhibitor will actually bind an allosteric site. So that's somewhere else on the enzyme that is not the active site. So it binds somewhere else on the enzyme. And by binding somewhere else on the enzyme, it changes how the proteins are folded in the enzyme. So the proteins kind of shift in order to accommodate for the binding of the inhibitor, and this changes the shape of the active site. Look at this before and after that non-competitive inhibitor bound the allosteric site. This is no longer the right-shaped seat for our substrate to sit, so this also makes this enzyme completely ineffective. And finally, we have feedback inhibition, another way to inhibit enzymes. And this is really just saying that the end product of a pathway can also serve as an inhibitor for the initial enzyme. So you can see this enzyme binds this substrate. It goes through this process and creates this end product. This end product can then bind the initial enzyme allosterically, so not in the active site, but it binds somewhere else on the enzyme, changes the shape of the active site, and now the substrate can no longer bind. This is a really important mechanism for allosteric regulation in cells, so regulating how much end product is made. So if there's enough end product made and the cell has plenty of it, it will shut down that pathway and not let any more substrate bind. Now we're going to do a quick segue into another topic. So when we're talking about metabolism, we're really talking about respiration. We're talking about anaerobic and aerobic respiration. So oxygen and metabolism go hand in hand. And oxygen is really important, or is it? So we're going to see that not all microorganisms need oxygen like we do. So microorganisms can live and thrive all over the earth in all of these different ecosystems. And so it makes sense that they have these different ways to deal with oxygen. Some of them need it, some of them don't, because all of these different environments are very different in terms of their oxygen concentration. So these are the different types of microbes in terms of their oxygen needs. First, we have obligate aerobes, and so they need oxygen to stay alive. We, humans, are also obligate aerobes. We need oxygen in order to make ATP or energy. Obligate anaerobes die in the presence of oxygen. Oxygen is actually poisonous to them. Facultative anaerobes, these aren't strict aerobes or anaerobes. They can survive in the presence of oxygen, or they can survive without oxygen and go through the fermentation pathway. We have microaerophilic bacteria. They require a lower level of oxygen than is just in the atmosphere. And then aerotolerant anaerobes. So these are anaerobes, but they can tolerate a very small amount of oxygen because they're able to detoxify those poisonous oxygen forms. And what do I mean by poisonous oxygen forms? So any organism that goes through the aerobic pathway, like us, have to be able to handle these toxic oxygen derivatives. So we'll see as we go through the metabolic process and use oxygen, you end up getting these superoxide anions, so these very negatively charged oxygens. These are not good. They can run around and bump into things and wreak havoc. So we need a way to regulate those and neutralize them. So that is through the form of enzymes. Look at this. Let me just point this out right here. Catalase, peroxidase, superoxide dismutase. Look at that. They all end in ase, and these are all enzymes that have the job of neutralizing these toxic oxygen species. So in the terms of superoxide dismutase, this enzyme is in charge of converting those superoxide anions into hydrogen peroxide. And hydrogen peroxide is a lot less toxic, but it's still kind of toxic. And whenever we are in the lab, we're really going to focus on catalase. So catalase is an enzyme that converts hydrogen peroxide into oxygen and water. And now look how neutralized that is. Everyone loves oxygen and water. In the lab, we're going to be able to identify the oxygen needs of an organism by growing them in special tubes called thioglycolate tubes, or TGA. And this just shows you those different oxygen requirements and what they would look like in the tube. So a thioglycolate tube is really cool because it's kind of a thinner auger than you'll be used to seeing, but this thinner auger has an oxygen gradient. So there's a lot of oxygen at the top where it's exposed to the air, and then there's no oxygen at the bottom. So whenever you inoculate this tube and bacteria grows in it, wherever the bacteria grows, which is all these black dots, will tell you the oxygen requirements of the organism. So this one's an obligate aerobe. All the bacteria grew at the top where oxygen was. This is an obligate anaerobe. All the bacteria grew at the bottom where there was no oxygen. Facultative, they'll use oxygen if it's present, but they don't need it, so most bacteria will be at the top, but there will also be bacteria throughout the tube. Microaerophilic, so they require a lower level of oxygen, and you'll see all of that growth kind of right below the opening of the tube. And then also aerotolerant bacteria, so they're not really affected by oxygen, and they'll grow evenly throughout the tube. So now we've talked so much about energy. Now we're finally going to get to the point where we talk about getting that energy out of glucose and what is that energy. We'll see that the energy currency of the cell, or the ability for the cell to do work, is in this molecule called ATP, adenosine triphosphate. So whenever you see ATP, think energy. ATP is produced during photosynthesis and cellular respiration. So whenever we're going through these metabolic processes, we're creating ATP and we're making energy for this cell by breaking down bonds. And we're creating ATP through a process called phosphorylation. So we're adding a phosphate group. So this is ADP, adenosine diphosphate, has 2 phosphates. We added a phosphate, and now it's so energized. It has 3 phosphates. It's adenosine triphosphate now, a very energized molecule. How are we getting energy from glucose? This is going to be through a process called carbohydrate catabolism. And glucose is the most common carbohydrate used in this process in order to get energy out of it. And glucose is catabolized. Oh, by the way, does anyone remember what catabolized means? Is it building or breaking? It has cat in it. Cats break things. So catabolism is the breakdown of a complex molecule. So glucose is being broken down to get energy out of the bonds. So glucose can be catabolized or broken down through aerobic cellular respiration or anaerobic respiration and fermentation. Aerobic requires oxygen. Anaerobic and fermentation do not require oxygen. Aerobic results in the complete breakdown of glucose, so we're getting so much energy out of it. So you get a lot of ATP. Anaerobic respiration and fermentation only partially break down glucose, so you get much less ATP from these processes. Now let's do a deeper dive into aerobic respiration. So we're going to start with a process called glycolysis. Then we're going to go visit the Krebs cycle. And then we're going to go to the electron transport chain. So this is the whole metabolic journey that we're going to go on in order to get ATP from breaking down that molecule of glucose. So step 1 on our journey of carbohydrate catabolism and breaking down glucose to get all the energy, we have glycolysis. And glycolysis occurs in the cytoplasm. And glycolysis, whether it's aerobic or anaerobic, you need oxygen or you don't need oxygen, either way, glycolysis does not require oxygen. So this one part of the process does not require oxygen. During glycolysis, we start with a 6-carbon glucose. So glucose has 6 carbons. By the end of it, we're going to end up with 2 3-carbon molecules of pyruvic acid, and that's what's going to go into the next step of the Krebs cycle. Also, by the end, we'll have a net gain of 2 ATP. So these next couple slides, I know might look a little intimidating or overwhelming, but don't worry about it because I'm not going to ask you about specific steps of glycolysis or to name certain molecules or anything like that. This is just to help show you that you start with a molecule of glucose that has 6 carbons and you end up with 2 molecules of pyruvic acid that each have 3 carbons. But the steps of glycolysis, it has these 10 steps, and the first 5 steps you have to invest energy. So you put energy in to start breaking down glucose. And then the next part of the process is the energy yielding phase. So you're getting energy out of breaking down glucose now. And you can see through all of these rearrangements, we actually end up with 2 molecules, 1, 2. Each one has 3 carbons, and each one is pyruvic acid. And this is the molecule that's going to go on to the next stage and move on towards the Krebs cycle. Before we can get to the Krebs cycle, though, we have to do a little altering with the pyruvic acid. So pyruvic acid can't go straight to the Krebs cycle. It first has to be synthesized into acetyl-CoA. So the synthesis of acetyl-CoA, we're going to end up with 2 molecules of acetyl-CoA, which makes sense because we started with 2 molecules of pyruvic acid. We're also going to get 2 molecules of CO2 out. So this is the carbon dioxide that's actually generated whenever you breathe out and you're getting rid of your carbon dioxide waste. It's from this process right here. And we're also going to get 2 molecules of NADH. Oh, look, an electron carrier. And it has a hydrogen. So this is the reduced form. It gained electrons. It's carrying electrons. And we're going to see later where this electron carrier is taking those electrons. So wonderful, now we have converted our pyruvic acid into acetyl-CoA, and now that acetyl-CoA can enter the Krebs cycle, or the citric acid cycle. It means the same thing. And there's a lot of energy in this acetyl-CoA. So remember, we started with a molecule, glucose, with 6 carbons. We got 3 carbon molecules of pyruvic acid, and now we have 2 molecules of acetyl-CoA that are going to go into the Krebs cycle. And as this acetyl-CoA goes through the Krebs cycle, energy stored in acetyl-CoA is being transferred to electron carriers, NAD+ and FAD. So these electron carriers are going to pick up electrons or energy from acetyl-CoA and take it somewhere else further down the metabolic pathway, as we'll see later. And the Krebs cycle or citric acid cycle occurs in the cytoplasm of prokaryotes and the mitochondria in eukaryotes. Remember, prokaryotes do not have mitochondria. They do not have membrane-bound organelles. So all of this is occurring in their cytoplasm. But for eukaryotes, this is taking place in the mitochondria. So the major function of the Krebs cycle is to reduce coenzymes or to reduce electron carriers. So we're giving electrons to electron carriers. As we're breaking apart acetyl-CoA, breaking bonds, that potential energy is being stored by electron carriers and being taken further down the metabolic process pathway. Remember that before we could go into the Krebs cycle, we had to convert pyruvic acid into acetyl-CoA, and CO2 was released. And then going through the Krebs cycle one time, we're going to get out 3 molecules of NADH, and this is reduced. It gained electrons in the form of hydrogen. And 1 molecule of FADH2, and this is also reduced. It gained hydrogens. So think about this. How many times will we go through the Krebs cycle for 1 molecule of glucose? And remember, 1 molecule of glucose has 6 carbons. So for 1 molecule of glucose with 6 carbons, we end up going through glycolysis and getting 2 molecules of pyruvate. Each one has 3 carbons. Then that's converted to acetyl-CoA, so we have 2 molecules of acetyl-CoA. And that means for 1 molecule of glucose, we're going through the Krebs cycle 2 times. So for 1 molecule of glucose, we would get 6 molecules of NADH and 2 molecules of FADH2. So like before with glycolysis, I will not be asking you anything about specific steps in the Krebs cycle here with names of molecules or anything like that. I just want you to know that the main purpose is to reduce those electron carriers. So we have NADH, 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 and then we have FADH2 right here. So the whole point of this cycle is to take acetyl-CoA, which has all this energy stored in these bonds, rearrange it, and go through the whole Krebs cycle, and then from those rearrangements, we're releasing energy from the bonds, and electron carriers are taking that energy further down the metabolic pathway. And that's the big takeaway here. That's really all you have to know from this diagram here. We're not going to go too in depth with this. So the Krebs cycle is complete. You went through a whole molecule of glucose, so you went through the Krebs cycle twice with 2 molecules of acetyl-CoA, and you ended up with 2 molecules of ATP, 2 molecules of FADH2, 6 molecules of NADH, and 4 molecules of CO2. This is also generating that carbon dioxide that you breathe out. So I've talked a lot about energy moving through the metabolic process and reducing these electron carriers, and they're carrying energy somewhere, so you should be wondering where are they taking their energy to? And the answer is the electron transport chain. So that's where all of those reduced coenzymes or reduced electron carriers are ultimately taking their energy in the form of electrons. In eukaryotes, the electron transport chain is going to be in the membrane of the mitochondria, and in prokaryotes, it's going to be in the cell membrane. So the electron transport chain is always associated with a membrane because embedded in this membrane are a system of these linked proteins called the electron transport chain. So this is where these electron carriers, NADH and FADH2, are going to pass their electrons into this system. The electrons are going to be transferred in the form of hydrogen ions down these different complexes or carriers. Eventually, the electrons are going to be passed to oxygen. So oxygen is the final electron acceptor for aerobic respiration. And then we'll see how at this point this generates a lot of ATP. So most of the ATP for cellular respiration comes from the stepwise release of energy through these different redox reactions, through the electron transport chain, through all those different complexes within the membrane. So there's two major events that are happening in order to get ATP while we're passing those electrons or hydrogens through the complex. So first off is oxidation reduction reactions. So electron carriers, NADH and FADH2, bring their electrons and protons to the electron transport chain. They pass them off to the first complex. And then these carrier molecules or complexes within the membrane pass electrons from one to another and ultimately to that oxygen, which is the final electron acceptor. Electrons are being passed along these different complexes in order to form a proton gradient. So that's the essential process involved in terms of transferring this energy from one complex to the next, to the next, to the next. So energy from each electron is being passed down this chain, and as the electrons being passed down, protons or hydrogens are being pumped from one side of the membrane to the other. So that's why it's important this is occurring in a membrane, because we're creating a concentration gradient where one side of the membrane is going to have a much higher concentration of protons than the other. This proton gradient gives potential energy. So as these hydrogen ions then flow back in order to correct the proton gradient, they're going to flow through this final protein right here called ATP synthase. Hydrogens are going to fly through here to fix the gradient, and that is what is going to give the energy necessary to turn ADP into ATP. So a phosphate is going to be stuck to ADP by creating all of this potential energy with this proton gradient. Another image on the electron transport chain, and I like this one because it really shows you the difference between a prokaryote and a eukaryote. In prokaryotes, the electron transport chain is occurring in the cell membrane, and in eukaryotes, it's occurring in the mitochondria. And as you can see here, we have these electron carriers, NADH and FADH2. They have electrons that they brought to the electron transport chain. They passed it off to the complex within the membrane. And then these electrons are going to be passed along from complex to complex to complex. And as that's happening, hydrogens are being pumped to the other side of the membrane. And that's creating a gradient, a proton gradient. So there are a bunch of protons on this side and not so many on this side. And that really makes protons want to get back to the other side and correct the gradient. And what they're going to do is rush through this ATP synthase enzyme. ATP synthase, ACE, it's an enzyme. And what this enzyme does, it uses this energy that it collects from hydrogen rushing through it in order to stick a phosphate molecule onto ADP, making it ATP, which is the energy currency of the cell. And you'll see here we have all these electrons moving around, but now that they've moved through the complexes, what are we going to do with them? Now comes in the final electron acceptor for aerobic respiration is oxygen. Oxygen will accept those electrons, resulting in water. So that is aerobic cellular respiration. That is going through those processes of glycolysis and the Krebs cycle and the electron transport chain with oxygen as the final electron acceptor, because we're talking about aerobic. And the whole point of this process is to make ATP, so it's to make energy so the cell can do work. And from aerobic respiration, we get a lot of energy or a lot of ATP. So you get about 36 molecules of ATP from one molecule of glucose. And we'll see the other types of respiration. So we'll talk about anaerobic respiration and fermentation and see that those are just not as energetically favorable. So now we'll talk about anaerobic respiration and fermentation and how cells get energy if there's no oxygen available. So if oxygen is not the terminal or final electron acceptor, how are organisms getting energy? So for anaerobic respiration, oxygen is no longer the final electron acceptor. There are lots of anaerobic bacteria that can still make energy, or ATP, using something other than oxygen though. So they can use nitrate, they can use sulfate, they can even use carbon dioxide. But anaerobic respiration creates less energy or makes less ATP because the full length of the electron transport chain is not used. So that's why less ATP is produced. When organisms use carbon dioxide as their final electron acceptor, and this depends on the organism, but they'll produce either methane or acetic acid. And this is just occurring naturally by bacteria in your gut, the guts of cows, and naturally in swamps as well. Now, there are organisms that can still make energy without the presence of oxygen and without even the presence of a final electron acceptor. So when a final electron acceptor is just not present, you can't go through the electron transport chain, but you can still go through glycolysis. So fermentation is this kind of alternative system that allows you to go through glycolysis without going through the other steps of respiration. And the main function here is going to be oxidizing NADH into NAD+, and then that electron is going to be donated to some other organic molecule. We'll see that on the next slide. And this is definitely not as energetically efficient. So from one molecule of glucose, we produce 2 ATP. Think back to aerobic respiration. From one molecule of glucose, we got about 36 ATP. So this is not as efficient, but still very useful for organisms to be able to make energy without the presence of oxygen or a final electron acceptor. So fermentation, we don't have oxygen and we don't have a final electron acceptor, so we're only going through the process of glycolysis. And if you remember, glycolysis produces 2 molecules of pyruvic acid from 1 molecule of glucose. And from this pyruvic acid molecule, depending on the organism, there are 2 different pathways for fermentation. There's acidic fermentation, and then there's alcoholic fermentation. For acidic fermentation, pyruvic acid is converted into lactic acid. And lots of different types of bacteria do this. Example would be lactobacillus bacteria. This is an organism that can cause milk to sour, and it can produce some acidic dairy products like buttermilk, or it's really useful for producing yogurt as well. And then we can also go through this other pathway, and this is the alcoholic fermentation pathway. And this is performed by yeast cells. So yeast converts pyruvic acid into alcohol or ethanol, and in the process, CO2 is also produced. So this is the process that's responsible for making things like wine and beer. And this CO2, this production of gas, is responsible for the presence of bubbles in like beer and champagne. So comparing fermentation and respiration, and this is talking about both aerobic and anaerobic respiration here. Fermentation, no electron transport chain is used. There's no final electron acceptor, so we can't use the electron transport chain. ATP is produced by the substrate level phosphorylation, and this means that we're essentially just not using the electron transport chain. We're only going through the process of glycolysis and making those 2 molecules of pyruvic acid. Respiration, on the other hand, uses the electron transport chain. Aerobic respiration uses all of the electron transport chain, and oxygen is the final electron acceptor. And then for anaerobic respiration, the whole electron transport chain is not used, and oxygen is not the final electron acceptor. For both cases, though, most ATP is produced through the electron transport chain. So how much ATP is being produced by these different processes? So through fermentation, which only goes through glycolysis, we're only making 2 ATP per glucose molecule. Compared to aerobic respiration, we're making 36 ATP molecules per glucose molecule. And of course, this yield is going to depend on the electron transport system. So for anaerobic respiration, depending on what is being used as the final electron acceptor, will adjust how much ATP is made through anaerobic respiration, but it's less than 36 per glucose molecule. It's not as energy efficient because it's not using the full electron transport chain. This final thing is just the Pasteur effect. So this is a regulatory phenomenon where microbes will shift from using fermentation or aerobic respiration depending on the condition. So we see that aerobic respiration is much more favorable, right? You get a lot of energy. It's fantastic. So that means if a microbe has an opportunity to go through aerobic respiration and make a lot of energy, it will choose to do that. It will shift and start going through the fermentation process if there's no oxygen available, but it will definitely prefer the more high energy option. Based on that information about the Pasteur effect, what do you think about this question? So you're making beer and your yeast are growing really well, fantastic, but they're not producing any ethanol or alcohol. So what is most likely the issue here? What should you change? And you can think about this for a second and pause, because here's the answer. So there is probably too much oxygen available for your yeast, and they're choosing to go through the aerobic respiration process to make a lot of energy, and they're not forced to go through the fermentation process yet. So you have to close off oxygen, you have to shut off oxygen access from that yeast and force them to go through the fermentation process, which will make them go through glycolysis to make pyruvic acid, which will then be converted into ethanol.